We'll take a simplified tour through the first 3 weeks of embryology. Each day finishes with some etymology because understanding of the language behind embryology is crucial to comprehending the topic well. Day 1. The egg is invaded by a sperm, together forming the zygote. It contains male and female pronuclei. Its cytoplasm is surrounded by a tough glycoprotein shell. Zygote means two things joined in a close relationship because sperm and egg have joined to become one cell. Day 2. The first mitotic division of the zygote occurs around 30 hours post-conception, producing 2 blastomeres. Blastomere means sprout segment because one blastomere is one segment of the developing human sprout. Day 3. Division continues, and once 16 blastomeres are present, they are together known as the morula, which means mulberry, because this bunch of cells visually resembles a mulberry. Day 4. Fluid enters the morula, forming an internal cavity. The morula is now known as blastocyst. From now on, we'll stop drawing individual cells and we'll just show the overall shapes of structures. Blastocyst, again sprout, and cyst just means small fluid-filled sac. Day 5. The blastocyst has shed its outer shell. Cells have separated into 2 distinct areas: the trophoblast, which develops into the placenta and will feed the embryo, and the inner cell mass, which develops into the embryo proper. Trophoblast comes from trophein, meaning to feed, because the trophoblast becomes a placenta which feeds the developing embryo. Day 6. Trophoblast cells break down barriers in the uterine epithelium, allowing entry of the blastocyst. Day 7. Meanwhile, the trophoblast begins to transform into 2 separate parts: cytotrophoblast on the inside, syncytiotrophoblast on the outside. The latter is one cell with many nuclei. Syncytiotrophoblast is appropriate as a name because it is not many separate cells, it is one together cell. To simplify things and match the imagery in most textbooks, we'll remove the endometrium now and rotate our view like so. Day 8. The inner cell mass differentiates into hyperblast and epiblast. A space develops in the epiblast, the amniotic cavity. Hypoblast meaning lower sprout, epiblast meaning upper sprout, and amniotic comes from amnion, which is Latin for membrane around a fetus. Day 9. Meanwhile, cells of the hypoblast have migrated to replace the blastocyst cavity with the primitive yolk sac. Hypoblast and epiblast together are here known as the bilaminar disc. Bilaminar means 2-layered. Day 10. The blastocyst is now firmly implanted in the endometrium. The syncytiotrophoblast breaks into maternal capillaries known as sinusoids, effectively co-opting their blood for the developing embryo. Day 11. On the inner surface of the cytotrophoblast, tissue known as extraembryonic mesoderm has developed, in orange here. This eventually develops cavities which form the extraembryonic coelom. Coelom is sort of another word for cavity. The result is extraembryonic mesoderm adhering to cytotrophoblast and amniotic sac, where it is known as somatopleuric mesoderm, and to the yolk sac, where it is known as splanchnopluric. Splanchnopluric basically means on the side of the visceral organs because this mesoderm adheres to the viscera side, the yolk sac eventually becoming the gastrointestinal system. And somatopleuric means on the side of the body. Day 12. The yolk sac pinches off part of itself, leaving us with a remnant of the primitive yolk sac, the exocoelomic cyst, and the secondary yolk sac, which we'll from now on just call the yolk sac. Exocoelomic cyst means fluid-filled sac outside the main cavity. The main cavity being what we will now call the yolk sac. Day 13. The space known as the extraembryonic coelom has expanded and is now known as the chorionic cavity. The extraembryonic mesoderm linking the cytotrophoblast to the embryo is known as the connecting stalk, which will eventually become the umbilical cord. Chorionic comes from chorion, meaning outer membrane of the fetus. Day 14, taking a closer look at the bilaminar disc now. Cranial just means head end of the embryo, caudal means tail end. We can see here this little dip in the middle. Cells in the epiblast begin to differentiate, change type, and burrow underneath the layer of the epiblast. So this looks like a burrow down the caudal end of the epiblast. It's known as the primitive streak, and seeing that marks the beginning of gastrulation. Gastrulation means becoming a gastrula, which is a trilaminar embryonic disc, and more on that in week 3. We'll bring in a slightly more detailed version of the bilaminar disc now. Day 15. At the cranial end of the embryo, there's a patch of columnar cells known as the procaudal plate. At the cranial end of the primitive streak, cells are burrowing thickly underneath the epiblast, forming, as they do so, this circular depression known as the primitive node. Day 16. By now, there are thousands of cells in the epiblast layer alone. It is the mass movement of these cells that produces these changes in the shape of the epiblast. The cells that burrow through the primitive streak and the primitive node differentiate into cell populations known as germ cell layers. These are progenitor cells, which go on to differentiate into the bulk of our bodily constituents. The germ layers are endoderm, in a darker shade of yellow here, and these cells displace and replace cells of the hypoblast. We have the germ layer known as mesoderm, and these cells push in between the layers of the former bilaminar disc. Some mesoderm spreads to the edges to meet the extraembryonic mesoderm. So these are now continuous. Epiblast cells differentiate into ectoderm, the third and most superior germ cell layer. The word germ literally means bud, seed, or embryo. Endoderm means internal skin, mesoderm, middle skin, ectoderm, outer skin. And the translation skin sort of makes sense given they are very thin layers to begin with. They look like individual skins. Day 17. By this stage, cells burrowing down the primitive node have started to form a hollow rod called the notochordal process. Notochordal is from noto, which means back, and cord, meaning cord, because the back cord, aka the notochord, later serves as integral structural support for the early embryo. Day 18. Ectoderm between the primitive node and the cranial end of the embryo begins to thicken and is known as the neural plate. Seeing this is the beginning of another process called neurulation. We have a separate video on that if you'd like to know more about it. Day 19. The notochordal process partly fuses with the endoderm now and is known as the notochordal plate. Given that the notochordal process was hollow, this means that now there's a passage between the amniotic cavity and the yolk sac, from the primitive node down through the endoderm. And this passage is only temporary, and it's known as the neuroenteric canal. It's thought to be important in equilibrating pressure between these two spaces. Our arrow travels through the notochordal plate into the yolk sac. Day 20. The neural plate has begun buckling in the midline. We call this dip the neural groove, and the mounds either side the neural folds. Day 21. These folds progress toward each other and begin to fuse in the centre around day 21. Where they have fused, they form a hollow cylinder underneath, which is the neural tube, the very beginning of the central nervous system. The notochordal plate has by now lengthened. It's closed its inferior opening, which was the neuroenteric canal, and it's now become a solid rod, which is the notochord. Just beneath the developing neural tube lies this structural backbone of the embryo. And that's all we'll cover for the first 3 weeks. As I mentioned